During sexual reproduction in multicellular organisms, sperm and egg join together to form a single cell with a set of chromosomes from each parent. In this example, we see a single fertilized frog egg as it divides over and over again. One would think this process would simply produce identical copies of the original cell, such as we saw when the amoeba divided by binary fission. Instead, what happens is that cells with quite different characteristics begin to appear after only a few divisions. By the time a free-living creature such as a tadpole is formed, it is made up of literally hundreds of types of specialized cells joined together in specific ways to create tissues and organs, all of which originated from the single cell of the fertilized egg. The method by which this process, called cellular differentiation, takes place is not well understood, but it appears to be the result of certain genes being turned on at specific times. Genes are the sites on DNA molecules that control the production of specific proteins, and as a result, determine the unique characteristics of cells. For example, a cell destined to become skeletal muscle receives chemical signals from the genes in the DNA, telling it to produce the unique muscle proteins actin and myosin. Other information is also provided so that the cell ends up in its distinct elongated shape. Each cell in the human body contains thousands of genes, but at any given time, only a relatively small percentage of these genes are active. The evolution of cells can thus be traced back to gene function. Simple cells have fewer genes and are incapable of the complex feats of cellular differentiation seen in multicellular organisms. And yet, they share many basic characteristics.